So hi everyone, I'm Cristina Fernandez. I'm one of the educators here at Cold Spring Harbor Laboratory DNA Learning Center. And we are here today to listen about our new speaker for Meet the Scientist series. We have Dr. Jessica Tollkuhn here with us to share about her path to become a scientist and also her interest in research at the lab. But before giving to her the power to start this talk, I'm going to just share with you guys a couple of things about our webpage. So we have here the Meet the Scientist series with her, and we already have the next talk that is going to be on April 21st with Dr. Ulas Petmal. He's a plant biologist, and he's going to talk about his research also at the lab. So you are able to register now and just be ready for that talk that is coming. We also have a lot of really interesting talks from before that they are both available through our webpage and in our YouTube channel. So you guys can go and watch them all. They're really interesting. And the second thing I want to mention is that we already have the registration open for our summer camps, our science summer camps. We have both virtual and in-person camps in our 3 different locations. The one here at Cold Spring Harbor in Long Island, the one in Brooklyn, in New York City, and the one in Sleepy Hollow, Westchester, Regeneron. So in Regeneron Pharmaceuticals, we have another location in which we're offering summer camps. So you guys are welcome to register or look at them and also share with other people that might be interested in that. If you are having financial needs, we also offer scholarships. So you can apply and let us know that you might be interested but struggle with that. So we'll be happy to help you. And now I'm going to pass it to Dr. Jessica Tollkuhn. And if you have some questions, you can put it on the Q&A section. And then at the end, she will be able to answer all of them. All right. Thank you, Jessica, for being here with us.
So thanks for having me. I'm going to talk about my research and looking at how hormones regulate sex differences in the brain. Let's see, what am I doing here? So just as a very quick summary, my lab studies how steroid hormones act on the brain. And so when I say steroid hormones, I actually mean a certain class of hormone. When people think about steroids, they often think about steroids that are actually like anabolic steroids, like for bodybuilding stuff, which is testosterone, or steroids. Often you can be given like prednisone or something like that as a prescription. That's also considered steroids. But like a steroid hormone is a certain structure of kind of a fatty hormone that's released at one place in your body and floats around and can act distally in other parts of the body, right? So steroid hormones I'm interested in are mostly sex steroid hormones that are made by the gonads, right? Mostly the testes and the ovaries. So we study estrogen, testosterone, and we do study cortisol, which is made in the adrenal glands a little bit. And we specifically study how these hormones act on the brain, right? So we know actually that hormones do a lot to sort of sculpt neurodevelopment. Hormones also regulate behavior. So certainly anecdotally, like there's relationships right between testosterone and aggression, or even with estrogen for women, periods when estrogen is high in the menstrual cycle or in an estrous cycle. If you're a mouse or a rat in the lab, when estrogen is high, it's actually lower anxiety. Having estrogen is good for mood. High estrogen is good for mood, and low estrogen increases anxiety and depression. We'll talk about that a little bit more, but this is very big picture of the kinds of questions that we're interested in. And we study these questions really with molecular biology and genomics approaches, and then also looking at neural pathways and behavior. So before I get into the details about that, I was just going to talk a little bit about myself and how I got here. I think this is a picture of me when I was 8 at a science museum with one of those Van de Graaff generator things where you can put your hands on, and the current, I don't know, I'm not even a physicist. I don't remember why my hair's sticking up everywhere of the charge. Actually, the high school students probably remember when my hair's sticking up, but I remember my hair sticking up in this picture. But I was always really interested in science. But going from like thinking stuff is cool and wanting to learn about things and having fun in science museums doesn't necessarily really tell you what it's like to be a scientist, right? And I think it took me a while. I just kind of kept doing science things, even in college, without really having a kind of a concrete foundation of what it meant. I think now students are much more informed about, or at least they're maybe forced into having to make life choices earlier about what they want to do. I knew I wanted to do science. I knew I wanted to major in biology. But I didn't really know what it meant. Like I didn't actually, I went to a small liberal arts school in Oakland. So I'm from California. I spent all my life in California before I came to Cold Spring Harbor as faculty. And I really liked my school, but it didn't have a lot of research opportunities. So I actually got most of my lab experience doing lab stuff at the bench, pipetting and learning molecular biology in industry, because I would get paid summer internships. And it was sort of like, in order to get experience, I had to be somewhere where there was something to do, but then I had to get paid enough to pay rent so I could keep going in. So I got at least paid working in industry in the summers enough that I could like share an apartment when I was in college in the summer and not go home where there were not as many jobs. So I got a PhD from UC San Diego. And then, you know, in biomedical sciences, you often, before you have a faculty position, you do a postdoctoral research, even more extended training. So my original background was in studying gene regulation and developmental biology. And then I got interested in trying to understand behavior. And so I went and did some neuroscience, and I will talk about connecting all of those together. So I guess, do I have another point after this? Yes, right. So here I am, I'm at Cold Spring Harbor, and I've merged these two interests of molecular biology and neuroscience. So I really like thinking about genes. I like thinking about how genes are turned on and off. I like thinking about how genes being turned on and off in a developing animal leads to like a wealth of interesting biology. And this is something that I just kind of found in undergrad that was something that really motivated me. And so going from, oh, these ideas are really cool to, oh, this is something that I can do and I can ask questions. And this, I think, transition point that you can, as a scientist, you get paid to just think about things that you think are interesting is just the best job to me. So I really enjoy being a scientist. I enjoy learning about science. I enjoy discovering new things in science. And it turns out that, you know, I had pretty good hands so I could do the experiments, but now I'm a professor. I don't really do much of the experiments anymore. I write the grants and the papers, and I mentor people in the lab and teach them how to do experiments and work together. And that's kind of really what being a professor is, even somewhere like Cold Spring Harbor, where we don't have undergraduates and you're not teaching a lot of classes all day, like you would be at, you know, most universities. Okay, so I'm going to talk about, first I'm going to talk mostly about kind of sex differences and sex variable biology, just to give some big picture background here. It's really important when you're doing research to consider whether you're thinking about things like you're developing a new medication for people, or if you're just working in the lab, to just think about people or model organisms or things coming from different backgrounds and different places. And so one of the problems in biomedical research historically is that the diversity of the research subjects was really low, and a lot of medications were developed only using men as test subjects, like kind of these cohorts of white men, or that people talk about this in like psychology a lot of the time too, that a lot of fundamental concepts in psychology, I totally can't think of a real example right now. It's like people were doing these studies mostly on white male college undergrads, because those were the research subjects that people had access to like do all of these things and develop these theories. But in this case, medication was developed mostly with men. And there's a problem with this. And there's a good example of this problem is that if you only use certain populations of people when you're developing something, then you miss cases where what you've developed might not apply to every group. And so the example that I'm showing here is, or referring to, is for Ambien, which is people take Ambien for sleep. And what they found when they actually started selling it is that women were taking it to fall asleep at night and then like getting up and driving to work the next day and falling asleep on the way to work, because when they hadn't used women when they were testing Ambien, and they basically, this was just a simple thing that like on average, women are smaller than men, and they were overdosing a subset of women. And they hadn't even had women in the trials to figure this out until it was actually on the market, right? It's like completely egregious. How is this even possible? Women were not required to be included in clinical trials until 1993. And so I'm a basic scientist, and I study how biology works, and people hope, you know, can use that information to develop cures. But even in the lab, people didn't use female animals for studying science. It wasn't really required. It's still kind of not required, but it's highly recommended by funding agencies. If you write a grant and you say that you're going to study something that you have both male and female animals in your grant, but that rule didn't even exist until like 2016. There's also been some interesting discussion about people just prefer to use male animals when they do their research, because the assumption is that female animals are going to be more variable because they have an estrous cycle and their hormones will be fluctuating. And so that will lead to variability in the data. So there was this bias that people just wanted to study male animals because the data would be more consistent. And that's a really interesting bias, because this idea that hormones are making females more variable is not like exactly accurate. There are aspects that are variable, but males are also variable, and males are also variable because male levels of testosterone vary throughout the day and even seasonally. So I'll show that on the next slide, but like most of the research still is performed in male animals, but data from male and female animals are equally variable if you look at all data overall. Sex hormones themselves are variable in both sexes. So I was talking about the menstrual cycle, like estrogen will go up and down and progesterone will go up and down. But what people don't realize so much is testosterone also goes up and down. Testosterone levels are higher in the fall compared to the spring. On average, they're higher in the morning compared to the evening. And also testosterone is actually, it's not a steady state hormone. It's released in response to luteinizing hormone surges from the pituitary in this pulsatile fashion throughout the day, every few hours. So testosterone isn't just kind of, people know that like estrogen goes up and down over days, but people are less familiar with the idea that testosterone, just a normal biology in humans and in animals, has like, it'll spike and then it'll go down, and a few hours later it'll spike and it goes up and down all day. So there's a wide range of normal of these levels of hormones within and between sexes and genders. And really, this is something that vertebrates have evolved to provide behavioral plasticity, right? There are animals that have mating seasons or migration seasons. You don't want to, it's good to have the flexibility of adapting your behavior to fit with your environment, and hormones allow that sort of plasticity to happen because they change the intensity with which animals display certain behaviors. Oh yeah, the winner effect is a good example of this. So this is the idea that if two male mice have a fight, the one that wins is more likely to win the next fight. There's something like that. There's actually a surge of testosterone following the win, and it's thought to help the brain consolidate the memory of the win. But there's very interesting relationships between hormones and behavior. And also fatherhood studies have shown that men that spend more time around their babies when they're little, their average testosterone levels will drop for a while. And it's thought that this is actually making, animals less likely to go and mate or fight or like go do something else, but it keeps them around to be more engaged in the parental care. There's a lot of sex variability in the symptoms and the kind of etiology of different psychiatric and neurological conditions. So many people are maybe familiar with the idea that autism is diagnosed more often in boys than it is in girls. But then depression and anxiety are more common in women and girls after puberty. There's also Alzheimer's disease there's twice as many women as men with Alzheimer's and twice as many men as women with Parkinson's, right? So these things have been documented in the clinics. But what we study in the lab is how do hormones actually relate to sex differences in the brain and how the brain functions or in disease risk. So we study this stuff in the lab, in model organisms. We work in mice, but to kind of step away from people for a minute, right? We know if you want to study genes in the brain and how they relate to behavior, you want to be sure that you're working on a brain region, because the brain's very complicated, that you know that this part of the brain is related to the behavior that you're studying, that it's causally implicated, that this brain region, if you manipulate it, it will affect a certain behavior. And we know that these gonadal steroid hormones, right, testosterone, estrogen, and progesterone, they regulate sex variable biology in vertebrates, right? So we know that there's male and female typical patterns of sexual or mating behavior. We know that males tend to have territorial aggression more than females do. And then maternal care, there's a lot of species where only the moms are caring for the offspring, but there are many species, especially birds, with biparental care. Or even there's interesting species of frogs or seahorses or, where the dads are actually the primary caregiver, right? So there's a lot of variability across the animal kingdom in how these behaviors are displayed, or if they are sexually dimorphic, like displayed at different levels in males and in females. And one thing that we know is that sex hormones act in early life to set up sex differences in the brain. So the neural pathways that regulate these behaviors are slightly different in males and in females. Overall, like the brain is pretty much the same in males and females, but the sex differences are localized to pretty small, regulate these innate behaviors. And we also know that these differences are activated by sex-specific hormonal profiles in adult animals. And that means, right, if you remove the gonads in adult animals and you don't have estrogen or testosterone anymore, I always use the analogy of people taking their pets to the vet to have their pets fixed, because if you remove the gonads from your pet, it's because you're trying to decrease the display of these sexual and territorial, and also territory marking, right? Urine marking behaviors are a big one. Mostly in males. And if you decrease testosterone levels, that you can decrease this. I think especially in, and again, it varies by species, right? Like this works for cats better than it works for dogs. But in general, these hormones do something to initiate these behaviors. Animals do not engage in mating and aggression before puberty, right? The pups don't do these things, but the neural pathways that make these behaviors possible develop in early life. Right, so we work a lot on estrogen, and estrogen is made in the ovaries. It's actually made in other tissues as well in lower levels. It's very interesting, like estrogen can be made by fat cells, or it's made in the bones. Estrogen is required for bone health. And we know a lot about the different target tissues that estrogen can work on, and we specifically study what it does in the brain. We know estrogen is beneficial for brain health, so beneficial, like it can be neuroprotective. That increases sexual motivation, it improves mood, it improves memory. Women are more physically active at high estrogen phases of their cycle. There's a paper I was on last year that was written up in the New York Times about this. If anyone wants to Google estrogen and exercise, there's a really interesting story about this. And we know these things at a phenotypic level, that estrogen does these things. There's one important point that I want to make, is particularly in rodents. Estrogen is also important for the male brain. This is true in humans as well, but estrogen actually makes the brain male in rodents. I say rodents, I say mice and rats. We work with mice. A lot of scientists also work with rats. And this is because there's testosterone in the circulation of males. And this testosterone is actually converted directly into estrogen by this enzyme called aromatase. And actually, this is part of how estrogen is always made anyway. This is 17-beta estradiol, like the main estrogen in the body, is that all of these gonadal hormones are actually made from cholesterol. And there's this biosynthesis pathway, and these different hormones are made from this cholesterol precursor. But this is an obligate step in making estrogen. Is actually all estrogen is made from testosterone. And it gets converted right away in the ovaries. So there's certain populations of neurons in the brain that express aromatase. And so what happens is that the male brain has estrogen in the brain, but it's not very much. And so there's actually a testosterone surge on the day of birth. And there's also in humans, there's a second trimester testosterone surge. So there's actually elevated testosterone in fetal development during the second trimester of gestation. And in mice, there's a testosterone surge on the day of birth. And we found that we and others have shown that it's actually converted, the testosterone is converted to estrogen directly in the brain. And so it's actually the estrogen that is masculinizing and not the testosterone in the circulation for the brain. And so I can show you what I mean in some videos here, because I find videos are just much more memorable than words. We have a male and a female mouse, and they're different colors because it makes it easy to see them in the video. And this female has high estrogen and progesterone. She's in estrus. And females who have high estrogen and progesterone are sexually receptive, and they'll allow a male to mate with them. So, whoops, what? Sorry, started this going here. Where's my play button? There we go. So he goes over, he smells her, she's holding still, and she's letting him mate. If she actually wasn't in estrus, she would walk away. The sexual receptivity is very tightly coupled to fertility, so that animals aren't mating when they're not fertile. It's kind of a waste of time. Very nice evolutionary trick. This female also has high estrogen and progesterone. but we gave her estrogen during her first week of life. So we were mimicking this estrogen that's produced in the male brain by aromatase in response to this testosterone surge. And now when we give this female's high estrogen and progesterone, she actually attacks the male because her brain has been permanently masculinized by this transient exposure to estrogen. And that is necessary and sufficient for her to have male typical type social behaviors in adulthood. So this is a really to work in because the animal's doing something that it would never, females don't respond this way to male intruders. Only males have this territorial behavior. So the other important thing, right? So big points here. There's estrogen made locally in the brain from testosterone in males. And the other thing, this is what comes to my love here, is understanding genes and gene regulation, is that these steroid hormone receptors are actually, they're called nuclear receptors, and that they are inside the cell. They are in the cytoplasm and the nucleus. They're not in the membrane. And these hormones are also they're called nuclear receptors because they sit on the DNA in response to hormones. So I've schematized this here. If you think about these little hormones floating around in the circulation, they're this fatty structure, and they can just pass through the phospholipid bilayer of the cells, and they can bind their receptors. They just kind of float on in there. And when they bind estrogen receptor, they cause a change, and the estrogen receptors can come together and they sort of change their shape, and they sit right down on the DNA. And then they make RNA, right? This is transcription, as you make the RNA from the DNA. And so this is how what we are studying is like, what are these genes in the brain that are regulated by estrogen or testosterone or these other hormone receptors we care about? And we know a lot about the target genes of estrogen receptors. So what are the genes that are regulated by these receptors in the context of breast cancer? Because people for decades, a lot of, it's easier to study cancer because cancer cells grow all the time. That's what makes them cancer. And you can have these cancer cell lines where you can get a dish of cells and they'll keep growing, and you can study them and look at the genes and look at how they change. It's much harder to do this with a brain. Most of the brain doesn't have these hormone receptors. They're only in really localized populations. And so this is what our big breakthrough has been scientifically, is that we have identified are regulated by estrogen receptor in neurons for the first time. So this is something, we have all of these things that we know estrogen can do, but now we have this repertoire of like new genes that we've identified that people have not seen before, that we know our estrogen receptor is sitting on. So we now can actually look at how these genes that we've identified are contributing to these interesting aspects of biology that we know are modulated by estrogen. So kind of right, our big questions in the lab are, what are the genes regulated by estrogen in the brain? How does this early life signaling cause permanent changes in behavior? And more broadly, like, how do these genes mediate behavior or mood or disease risk? Are some of these genes protective, or do they increase susceptibility? Like, there's a whole theory about fluctuating hormone levels can predispose some people to depression or anxiety. And this is like, when women go on birth control, sometimes birth control improves your mood and sometimes it ruins your mood. And there's not a lot that's known about how changing hormone levels across different individuals will, to give them different side effects or not, right? We really don't know very much. We know that these things happen, but we don't know why. And those are one of the things that's what my lab is studying. Right, so we have all these new gene targets now that we found that we can see these genes that ER Alpha sits on. And we're interested in studying what these genes do, both in males and in females. So one of the things that we found is actually, if you remove the gonads, like the sex differences in the brain actually go away. The same genes will be expressed in males and females if you don't have any hormones. And if you give them the same hormone, if you give a male mouse and a female mouse the same amount of estrogen, the genes that are turned on in their brain are exactly the same. So really, if you see something that's different in the male brain or in the female brain, and again, we're looking in very specific brain regions here. Most of the brain doesn't have hormone receptors. But the sex differences that you can detect, they're variable. They're a readout of how the hormones look at the time. So if the hormones are going up and down, if you're looking at different parts of the lifespan, we're actually interested in aging now and when hormone levels drop, right? They drop in men too, they just drop more than menopause in women. But how does the gene expression change? in relationship to hormone levels changing, like across the estrous cycle or menstrual cycle. So we think that hormone responsive genes are maybe underlie sex differences in disease susceptibility, like I said, and we're trying to use these targets to understand resilience in both sexes. And now we're also interested in, like, we've done this with estrogen. We want to know what testosterone's doing. We want to know what the cortisol, the stress hormone, those hormone receptors are called glucocorticoid and mineralocorticoid receptors. And then I just have a couple more things I wanted to mention. We also have a program totally unrelated, not just, so I talked about like people not studying women and developing drugs when I was kind of how I framed the study of sex differences. But I'm also interested in species diversity, because things that we learn are also constrained by the model organism that you study. If you find something, you think, oh, this is how it is, but it's just, maybe that's just how mice are. And so we have these other species of rodents in the lab, and we have them in the lab because we're very interested in the social behavior of these prairie voles, because they may have lifelong pair bonds. So they pick an animal and they mate for life, and they're considered socially monogamous. And then the other species, the meadow vole, doesn't do this. So pair bonded voles have biparental care. The dads are really good dads. The older siblings will take care of the younger siblings. And we have a whole other research program to kind of study the evolution of social attachment and how the brain is able to form a social connection. And we're doing this, again, by taking the same kind of molecular approaches to look at the genes. In this case, instead of looking at sex differences, we're looking at brain regions that regulate social behavior, but we're looking for species differences. So we can understand what gives this prairie vole this unique ability to really form this adult attachment between two animals. And this has a lot of implications for psychiatric conditions where social interactions are kind of blunted, right? Whether it's autism or even people with schizophrenia or depression can become very withdrawn. And this isn't something you can study in mice, because mice, all mammals pretty much care for their offspring, but mice and rats don't care about each other in the same way. If you separate a bonded pair, they show depression and anxiety. They want to be with their partner. And so this is something in the lab that we can use to study, what that attachment really means and how it can form. So yeah, with that, I think I just want to acknowledge my lab members and my collaborators. So I have, 2 amazing graduate students in the lab and a postdoc. So this is someone that got their PhD, and then she actually got her PhD doing something very different than what we do. And then she's bringing her expertise to our lab. And then my lab manager, Melody Wu, so staff scientist who has a PhD, who's been working with us. She's not writing the grants, but she's doing like the really complicated experiments. So it's good to have, I think, people coming, continuing the kind of the diversity theme. Like, my student Kat came into the lab with a behavioral focus. She was really interested in behavior. And then Bruno came into the lab with a very molecular focus, and he was interested in the genes. And then Simone came into the lab, and her expertise is in neural physiology. And so bringing people together from different backgrounds, I think, makes people better at asking interesting questions in their experiments too.